spaces are the mesh. And when you go to process these for histology, you section them out, you actually put them in plastic and you degrade the mesh material, so you're left with the hole. And then you can see around the hole, which is just beautiful. The first thing, A, is H&E staining. It's a general cell staining. It stains the nucleus and the cytoplasm of the cell. But look at the response the body had to that. I mean, just attacked it, okay? So, Jeff talked about the fact that when you get such a host reaction to some sort of synthetic mesh implant, back in the day when they first designed these, they thought that would be great. Let's get a huge reaction to a hernia mesh and we're all set. Well, that's not the case. If you get a huge fibrotic reaction to a hernia mesh, not only are you going to cause contraction of that mesh, but you're going to cause adhesion to other organs. And so that can be very, very painful, and it can be deleterious to patients. So, and that's just a different staining right there. That's an antibody staining up there. And these are just fluorescent stainings of the levels of collagen and fibroblasts. This is great. So, I don't really like eyes. I mean, if they're nice eyes, I like them. But I don't like seeing anyone put their contact lens in their eyes. It totally skews me out, which is odd because I can take someone's guts falling on the floor, but I can't take that. But this is a really neat example of kind of in between a soft and hard tissue implant. So this is a cornea implant. Okay, so corneas are interesting. They're really, really interesting because they're very, very simple structures. They cover your eye. They're there to protect. They're there to refract light, but they're there mostly to protect because your lens refracts light as well. They're made of epithelial cells. So there's 3 layers. There's the epithelial. If this was the outside of the eye, the thin epithelial layer on the outside. Then you get this stromal matrix, which is made of very few cells, extracellular matrix, mostly water, 7% water. And then you get the inner layer, which is D-cement membrane, which is made up of, again, of epithelial cells, but they control diffusion of water and nutrients into the cornea. Right? When something goes wrong with D-cement membrane, it goes, and something goes awry with that diffusion of nutrients, water and particles can build up in the cornea and make it opaque. Right? Many times people have corneal injuries. Anybody ever get a stick in the eye or scrape their cornea? Okay. I ran into a tree playing tag. It was right behind me. I didn't see. I turned around, bam! It's like I knocked myself out. Scraped my cornea at the same time. I recovered. I was little. But if you have a trauma to the cornea and it doesn't heal, and you have herpes, that's just as a side. If you have herpes, you can't get these implants. It's just a funny little caveat there. It has to do with infection and all that. It's not just some random thing. But you might need a corneal implant. Now, there are corneal transplants, which are allogeneic from another individual. They can be transplanted. One person can donate 2 corneas, right? You don't really have to tissue type them that well because it's more with the site and all that. Diabetics it's the only thing they can donate are their corneas. This is not vascular, so that's good. But if you're not in line for transplant, you have to get a corneal implant. And what they do is, and I will go through this later in the semester, but the implantation is that they have to take out the natural cornea, make a little pocket, stick this thing in here, and they use sutures. So these are sutures that suture this polymethyl methacrylate, fairly hard but flexible disc to the rest of the cornea that remains here. And this is very important. You have to have integration of these fibroblasts from here integrating into the device to hold it down. What you don't want are the fibroblasts continuing to cover. All right, so that's one of the things. But it's important to understand that even though it's a fairly simple implantation, if the patient doesn't use their eye drops, it's so simple as eye drops, but if they are not compliant and they don't use their eye drops, they will reject the implant, okay? Because they have to keep it moist, they have to keep bacteria away from it, fungus, things like that. And so you can see here that there's a little bit of a reaction around here. You see a little bit of a reaction around here where you have kind of swelling of the material. You can kind of see in this picture, you can see the suture starting to bow a little bit, and that's because there's excess fluid and inflammation around that implant. What this leads to for this soft tissue, and I include this in soft tissue because polymethylene methacrylate can be in one form and injected. It can be processed so it's flexible. It's not truly hard, stiff material. It's flexible, so I put it in here. And then you can use it as glue, and it can become hard as rock. So again, different formulations of material. But what's important here is one of the biggest problems with these implants is that patients don't put the eye drops in. Okay, they don't comply, and that cornea, if you can see there, that implant starts to lift up off of the native tissue, and it extrudes, meaning it pops out. It's not like a person walking down the street, it pops out on the sidewalk and then people looking for it, okay? It'll slowly start to extrude out, and the sutures will break because they haven't completed degrading, and they'll snap. So it can be a big problem with that. So what do you do? And this is important. This is important for this lecture. It's going to be important for the whole class. Is that the implants we have now? They're fine. They're good. I mean, we're optimizing all the time. But if you want to make a really, really optimal implant that is biocompatible, and I said biocompatible before just meant benign. It didn't interact with the host, and if it did, it was negligible and didn't cause a big problem. Now we want interaction that's helpful. So that cornea implant, the greatest thing for a cornea implant would not only be to have, the outside is just meshed. It just has pores in it so the fibroblasts come in and anchor it down. It'd be nice to add a ligand onto that attracts only a certain amount of fibroblasts, only a certain cell type, and it stays there. I mean, that's the biologically active modifications we're talking about. So some of the modifications we're going to talk about as far as soft tissue and hard tissue as well are things like antimicrobial coatings. Did Jeff talk about that last week, antimicrobial coatings? Okay. They just have a new product out. I can tell you. I do writing for them, so I forget what to say. But, coating a surface with antibiotics. All right. Sounds simple. You have to think about, well, how are you going to link those antibiotics to that surface? What are you going to use? What sort of link are you going to use? Do you need an antibiotic? Can you put silver on a surface? So when we talk about titanium implants, when we talk about stainless steel alloy implants, why not coat them in silver? Silver in itself is antimicrobial. And so the orthopedic industry is looking at coating their implants with not so much an antibiotic coating, but an antimicrobial coating like silver. So that's one way to do it, to prevent infection, okay? It doesn't have anything to do with tissue integration. The next thing you could imagine is tissue integrating surfaces. So a long time ago, one of the PhDs in our lab was working on taking just the RGD group, which is a very simple amino acid sequence that is found in extracellular matrix. That's critical for cells to attach to each other into the matrix in the human body. Just isolating out those 3 amino acids and adhering them, processing it so that will stick to a biomaterial. And you take that biomaterial, implant it with the hopes that you direct which cells adhere. Now, if you're taking your surface and you're adding RGD groups, that decreases the surface area that other proteins have to attach, which is good because you're really directing your device. So that, that is still being worked out. It's very difficult to do technically. The idea works. In theory, it's just technically getting these on these devices. Contact killing surfaces. Say you don't want to elute an antibiotic. You don't want to coat with silver. You don't want anything being released from your device. But you want to make sure that if bacteria come in contact with your device, they'll be killed. Or they'll be sequestered so that antibodies can come in and get them, right? So find receptors specific for either portions of a bacterial cell surface or for certain immune cells or cytokines that are really there ready in case there is some sort of attack by bacteria. Another way of looking at it is you could take this and you could say, well, I'll think of contact killing in another way. I'm going to prevent cells, bacterial cells, from making biofilm. So again, Jeff talked about biofilm, and I talked a little bit about it. That's one of the biggest problems with the device industry now, no matter what device you're looking at, because bacteria, they interact with each other, all right, through that form sensing, like I said. And once one bacteria has interacted with these chemical signals with another, it turns on genes in that bacteria to produce the proteins that make the biofilm, right? Cut that communication off. If a bacteria doesn't have biofilm, they're not going to colonize and spread over the surface. So you can imagine putting molecules on the surface that disrupt that, right? And that might be a very easy thing to do. So you prevent that whole cascade of events from going on. And then there are multifunctional. You can imagine taking, this is a wish list here. You take all of those, you combine it all to make the perfect surface. But you have to remember, the body has a very ingenious way of getting around some of these things, and so does bacteria. So you have to be careful with things like antibiotic coating. You don't want to load a device with antibiotics and then have an antibiotic-resistant bacteria form on that device. Right? So you always have to think about the next step and the next step. So far, it's been shown that nothing forms a resistance or it's able to overcome silver. So, I mean, there are some metals that are still tried and true, they're still good. But when you're talking about chemical modification, the body has a great way of getting around it. So you have to be very careful, and coming down to multifunctional coatings could give you more problems than it's worth. Okay? So that's it for today. We're going to talk about, I believe I started on meniscus, or we do orthopedic stuff. And I love meniscus, when people just slam their meniscus. Because trying to fix that is very, very difficult. So there's a lot of really cool ways to do it. And we'll start that next week along with Lynn.